Hi, Rhonda. Thank you so much. Hi, everyone. My name is Ishani Shah, and I'm your local account manager at Advanced Cell Diagnostics. On the call, we also have your cell and gene therapy specialist at BIO-TECHNE, Loida Torres. And from the both of us, we just want to say thank you so much for joining us today for a joint webinar. A major focus of BIO-TECHNE and ACD has been to support our cell and gene therapy applications. Our innovative cell and gene therapy solutions are helping to solve various hurdles in this space, and this webinar is to highlight some of those tools. As Rhonda said, we have a very exciting presentation set up for you today in the cell and gene therapy space. And now I'd like to pass it over to Rhonda. Rhonda, back to you. Thank you.
Thank you very much, Ishani. That was great. And we're going to go ahead and ask the first speaker is Anushka Dikshit. She's one of our application specialists and has an excellent presentation for you. But here we go. So,
excellent. Okay, so welcome everyone, and thank you for this opportunity to let us talk about our unique technology and its applications specifically today in cell and gene therapy. So for those of you who are not familiar with our RNASCOPE platform, it's an in situ hybridization technology which was specifically designed to overcome some of the traditional limitations of an ISH assay, and while still giving you that high sensitivity and specificity of detection. So our technology is based on proprietary probe design strategy, strong signal amplification and background suppression, which ultimately allows you to visualize a molecule of RNA with single cell resolution. So the RNASCOPE technology has been developed as an ideal spatial analysis solution to interrogate complex tissues. Because it's so specific and sensitive, we can detect RNA biomarkers in cells, tissues with morphological context at single cell level. It consists of three major parts: the target probe design strategy, where ACD will design a specific target probe against the target transcript of your interest. Then we have the signal amplification system, where our special technology amplifies target gene signal and suppresses background noise, giving you high signal to noise ratio. And finally, our detection or quantification strategy, which allows you to visualize a single molecule of RNA as a punctate dot, as given here in the image, with single cell resolution, and sometimes even subcellular resolution. So because our assay is so specific, we wanted to make sure that it's flexible enough to be made applicable to different sample types and target types. So with our assay, you can detect mRNA targets, long non-coding RNA targets, splice variants, highly homologous sequences, and so on. And this assay can be applied to cells, FFPE tissues, fixed and fresh frozen tissues, PBMCs, and so forth. So not only is our assay sensitive and specific, but we have developed the assay so that it can expand to a number of sample types and target types as well. And I'll go in detail in some of the next few slides to expand on that a little more. First, I want to talk a little bit more about what makes our technology special. So the two key features of our technology are, as I mentioned, the probe design strategy and the signal amplification system. So here you can see that we have depicted our RNASCOPE probes as a Z molecule. And this is mainly to highlight the three different parts of the probe: the target binding site, where the sequence is such that it will specifically bind onto your mRNA target of interest. Then you have your pre-amplifier binding site, which forms the foundation of your signal amplification tree. And then finally, you have the linker region, which connects these two parts of the probe. So when we design a probe against a target of your interest, we design pooled probes of these specific target probes, and we design them such that they bind in pair to your target sequence. And this is especially important because the preamplifier, which forms the foundation of your amplification tree, only binds to a paired Z probe. Once the preamplifier is found, we have the amplifiers that bind onto the preamplifier. Then we have the labeled chromogenic or fluorescent probes that bind onto the tree, giving you high degree of sensitivity. So because of this strategy, we can detect targets that are really low expressing as well. And finally, we ensure specificity by making sure that if at all a single Z probe non-specifically binds to a sequence, the preamplifier will not be able to bind to this probe unless another Z probe is bound in tandem to it. So this ensures the specificity of our assay, giving you ultimately a very high signal to noise ratio. So this is the basis and foundation of our technology, which makes it extremely unique from some of the other tools and strategies that are available for spatial analysis. So just broadly under the RNASCOPE technology umbrella, currently we offer three main assays. The RNASCOPE assay, which is mainly for targets that are over 300 nucleotides long. Normally, the way we design probes is a pooled probes of 20 ZZ pairs. The BASESCOPE assay, which is our unique assay specifically designed for really short targets, which are less between 50 to 300 nucleotides. So these include your splice variants, your highly homologous sequences, and also point mutation detection. And finally, our recently launched microRNAscope assay, as the name suggests, was designed to detect targets that are really short, between 17 to 50 nucleotides long, and these are mainly your targets such as microRNAs, siRNAs, and antisense oligos. So these are our three major branches of assays under the RNAscope technology. Here in this slide, I have described the broad product portfolio that we offer with these three assays. As you can see, we have flexibility in our plexing capability. We have RNAscope assays that are single-plex, duplex, multiplex that can go up to 12-plex with the high-plex assay, where you can detect 12 targets on the same sample. Similarly, with BASESCOPE assay, we offer single-plex and duplex, and with microRNAscope assay, we currently offer single-plex. So our assay is also compatible with some of the automated platforms, as mentioned here, the Ventana system and the Leica automated platforms. If you need more information about which assay is compatible with what system, please don't hesitate to reach out to us. Currently, we have over 26,000 probes in our catalog, where they're already designed for specific targets. But if you're interested in a custom probe that is not in our catalog, then we can design a probe against any target and any species in as less as 2 weeks. So having established what our assay can do, let's actually go ahead and see what are the broad applications of this assay and focus on cell and gene therapy applications. So because of the flexibility that our assay offers, we have been used extensively in detecting all of these different key targets, which include GPCR, CDR3s, for spatial mapping, validation of single-cell sequencing, and so on. And the research areas that we have been used extensively include neuroscience, stem cell development, stem cell biology, developmental biology, for infectious disease research, especially in the current environment. We were able to design a probe against the SARS-CoV-2 virus in less than 2 weeks, which allowed for expedited research for our research community. Then for biopharma discovery and development, which is the topic for today, development of your cell and gene therapies that have shown considerable success in recent years. And finally, for cancer and clinical research. As I mentioned, today we'll be focusing on the applications of RNAscope assay for preclinical model for the study of preclinical models for cell and gene therapy to assess the biodistribution, safety, and toxicity of these cell and gene therapy drugs. So without delaying further, let's start with our first example, where I'm going to talk to you about how RNASCOPE was used for preclinical safety and activity assessment of CAR T cells. So this is a project that we did in collaboration with Bluebird Bio, where they were wanting to develop CAR T therapy against certain solid tumor antigens like BCMA and ROR1. And I'm going to talk to you about how RNASCOPE is used to help develop that CAR T therapy and assess the safety in the preclinical model. So they transduced CAR T cells in vitro and injected either anti-BCMA or anti-ROR1 CAR T cells in this xenograft mouse model. This mouse already had xenograft tumors, after which the treatment was injected. And the goal of the study was to assess how safe these CAR T cells were. What was the biodistribution of these CAR T cells like in the tumor? And was there an actual activation of the CAR T cells in presence of the antigen? So for this, we first designed probes against the BCMA and ROR1 antigen, which were the two antigens against which they developed the therapy. This allowed us to visualize these antigens within this xenograft tumor. Secondly, we wanted to assess whether these therapies have any non-specific effects, which in the cell and gene therapy terms, if they have any on-target on-tumor effect, or they have any on-target off-tumor effects, which can lead to severe toxicity. And finally, if these CAR T cells are actually engaging in presence of the activation, in the presence of the antigen, which tells us how effective they are in treating or against the specific antigen against which they are designed. So to do this, we designed probes, as I mentioned, against these antigens and also against the CAR T vector, the 3' UTR of the CAR T vector that was used to deliver these therapies. So now in this particular image, I'm showing you an example of how we looked at the expression of BCMA antigen in the mouse spleen and the mouse spleen on your the normal mouse spleen. I'm sorry. On your right hand side, you can see an image where we don't see any BCMA expression in the mouse spleen, as you would expect, since it's an antigen predominantly expressed in tumor cells. On your left-hand side panel, we see, using the CAR UTR probe, presence of the CAR T cells, which means that the CAR T cells are present in the spleen, but they are not engaging, which is indicated by negative staining for granazyme B. Granazyme B, as you know, is secreted by active T cells upon engagement. So absence of granazyme B shows that although the CAR T cells are present, they are not engaging. And this is exactly what you would expect. On the other hand, when you look at the actual xenograft tumor, which is positive for the BCMA antigen, indicated here in red, we see that the CAR T cells in red are present and are engaging, indicated by positive green signal by granazyme B. And this is exactly what you want, that in presence of your antigen, your CAR T cells are engaging and are active. So that tells you that the anti-BCMA therapy was pretty successful against the antigen for which it was designed. So the treatment was specific. Looking at the biodistribution, we see that it's only engaging in the tumor, which tells us that it is potentially effective and specific without any toxic effects. On the other hand, their second therapy, which was the anti-ROR1 therapy, had a different story to tell. So once this therapy was introduced in the mice, the researchers saw that the mice were showing some signs of toxicity. So upon sacrifice, when they performed IHC on the lung tissue of these mice, they were not able to see any ROR1 expression. But when they performed RNAscope, they were able to see that the lung of this mouse was actually expressing ROR1 to some extent. And this was causing the CAR T cells in the lung to engage and cause lung toxicity. Similarly, for the liver, they were able to see some hepatotoxicity. And when they looked closely with RNAscope, they saw ROR1 was expressed in the liver cells. As a result, they were engaging, CAR T cells were engaging and causing toxicity. So this really highlights how really basal level of antigen presence can engage these CAR T cells. So it's extremely important to assess how specific these treatments are and if there's going to be any toxicity. And sometimes assays like immunohistochemistry may not be able to tell you the complete story, as they might not be as sensitive. On the other hand, for the ROR1, when they looked at the tumor, they were not able to see a lot of engagement using the anti-ROR1 CAR T cells, telling them that this treatment was clearly not as effective as the anti-BCMA, and there's further research that needs to be done. But in this next image, I want to show you how using our multiplex assay, you can visualize multiple targets on the same section. So this is an image from the anti-BCMA CAR T study, and we have visualized the CAR T cells, granzyme B, and we have performed immunofluorescence for CD3. So with our RNAscope assays, a great advantage of this technology is that it can be combined with immunohistochemistry or immunofluorescence. And here's an example of that, where we have used the CD3 antibody to visualize T cells and then also identify the CAR T cells. Again, a same image now in the presence of interferon gamma with CAR T cells, which shows you which of these CAR T cells are active, and CD3 marks all T cells. Using our 4-plex assay now, here we are visualizing all of these 4 targets together, giving you a complete picture of how this tumor has the presence of its endogenous T cells, the CAR T cells, and which of these CAR T cells are actually engaging in presence of this antigen. So this is a very powerful study which told us how our technology can really help support the safety and efficacy studies of these engineered T cell therapies, which are showing considerable success at a preclinical level and shows the potential for actual therapeutic strategy for not only cancer, but some other pathologies as well. So when we actually assessed the tumor size, what we saw was that with the ROR1 therapy, there was clearly no change in the tumor volume, but with the anti-BCMA therapy, which we saw was very specific, we actually saw a reduction in tumor volume, suggesting that this is a very effective therapy and it is actually decreasing tumor size. So with the RNAscope technology, we were able to assess which of the tissues showed presence of the antigens against which we developed the therapy. We were able to design probes specifically against the CAR vector, identifying the engineered T cells from the endogenous T cells. We were also able to visualize the engagement of these T cells by looking at the soluble factors like granzyme B and interferon gamma. And ultimately, we were able to show the efficacy of these drugs because of the sensitive detection of RNAscope of the CAR T cells in vivo. So ultimately, with this technology, you can assess the efficacy and safety of your engineered T cells and understand if the biodistribution of these cells is as expected and if they're showing any on-target or tumor effects, which can be highly toxic. So this is an example of RNAscope technology being used in CAR T cell therapy applications. Next, I want to show you an example of gene therapy applications for RNAscope technology. Again, gene therapies are also showing a lot of potential in treating a number of disorders that were thought to be, as they call, undruggable. And a lot of these are eye-related disorders. So here's an example of how we use the AAV-based therapy to transduce monkey retinal cells. And an example here that we've used is that of an AAV transduced GFP vector. And in this particular example, we wanted to show the use of RNASCOPE in studying the AAV transduced cells. So we transduced part of the retina with the AAV vector, and part was a NAԻVE transduced region. And we wanted to compare the expression of the transgene and the vector between these two regions. So we designed an RNASCOPE probe against the promoter of this AAV vector and a probe against the actual transgene, which in this case was GFP. So here's an example where, as I mentioned, part of the retina was injected with the AAV therapy and part was NAIVE injected or injected with control. And as you can see, only the injected region shows the expression of the transgene and the vector using our RNASCOPE DUPLEX assay. So all these layers are your retinal layers, and you can see that in red is your GFP transgene, which is abundantly expressed in these different layers of the retina. But if you look closely, there is expression of, you can see the expression of the CB promoter in green, but the expression of the transgene is so high that in this image, the expression of the actual promoter is almost masked. But if you compare that to this NAIVE injected region, you see that there's no signal as expected. So this assay can really, again, help understand if your gene therapy is specific, if it's specifically targeting the cells and the region of the tissue of interest, and whether it's expressing the transgene to the degree as expected. Here is another example of the same region, a more zoomed in image, where you can now actually see the cells that are showing green positive signal, which actually tells you that these are the cells that have taken up the virus. And you see that some cells are expressing the AAV, while some are showing the presence of green signal, while some are not. And this is because these AAV therapies are known to show some degree of tropism when it comes to transduction. So some cells have taken up more virus than other cells and so forth. Again, the expression of the transgene, which is the GFP here, is shown in red. Another example showing very high expression of this transgene in this outer nuclear region compared to the NAIVE transduced retina. So in this particular example, the idea was to show that RNASCOPE can be successfully used to understand, again, the biodistribution of the AAV therapy, visualize the presence of the transgene, and understand whether it's transducing the specific cells of interest within the tissue of your interest. And because of the specificity and sensitivity, we can not only track the virus, but we can also, or viral vector, but also visualize the expression of the actual transgene. And on a similar note, I want to show you the application of our BASESCOPE assay, again in AAV therapy. Here is an example where this project was done by our PAS group, where they were testing, again, their AAV therapy in liver tissue, and they used our BASESCOPE DUPLEX assay to visualize this AAV therapy. And here, again, we designed probes against the actual AAV transgene in red and a probe against the actual AAV vector in green. So that again shows us which cells have taken up the virus and which cells are expressing the transgene. So in this particular example, I want to highlight how our technology can really give you not only single cell, but subcellular resolution, where you can, if you observe, the green signal is specifically in the nucleus, because this is the signal for the AAV vector genome, and the red signal is present in the nucleus as well as the cytoplasm, because this is your AAV transgene signal. So this really shows you specifically which cells within a tissue are expressing your transgene and which cells have taken up your virus. And here they compared it to a untreated or vehicle-treated control, and obviously the expression of the transgene is absent in the vehicle treated. So both our RNAscope and BASESCOPE assays can be used for AAV therapy analysis, and this has huge clinical relevance because in the recent years we have a lot of gene therapies that are being tested on a preclinical level for an ultimate clinical for the use of these treatments in clinic for undruggable disorders. Finally, I want to talk to you about our newest assay, which is the microRNAscope assay, which, as I mentioned, was specifically launched to detect really short targets between 17 to 50 nucleotides in length. And the targets that it detects are your siRNAs, mRNAs, and ASOs. So the antisense oligo it's not a new technology, but in the last few years, we've seen that it might have some real therapeutic potential, especially in the fields of neuroscience and oncology. Then you have your microRNAs, which are known to have pathological significance in a number of disorders. So in research to understand the expression pattern of microRNAs, you can use our new microRNAscope assay to study the different microRNAs. And siRNAs, again, are your synthetic oligos, which are used as therapeutics because of their specificity against the target of interest. So for microRNA applications, again, we have been used extensively in oncology, neuroscience, and immunology to identify microRNAs that are responsible for initiation of certain malignancies or for promotion of certain neurological disorders and so forth. Here's an example of how our microRNAscope assay was successfully used to visualize MIR124 across the brains of these different species. Again, this just shows you the specificity of our assay and how we can visualize punctate dots as positive signals. Next, we have designed probes against specific siRNAs. And again, this is a big biopharma application as a lot more companies are trying to derive siRNA-based therapies, again in oncology, to target specific oncogenes that might promote the malignant potential of certain cells. Then in neuroscience, to treat acute brain injuries and neurodegenerative disorders, and for autoimmune disorders to mitigate their progression. So here's an example of how one of our customers used our microRNAscope assay to detect their siRNAs within the tissue. Here we have our negative control, and here we have our siRNA1 that was delivered, and in this panel we have our siRNA2. Now, when we used a probe specifically against our siRNA1 therapy, we can only see positive signal in the siRNA1 treated tissue, showing you the specificity of our assay and how even for short targets, we can be extremely specific in designing our probes. And finally, similar to siRNAs, our other small oligotherapies, such as ASOs, can be assessed by using our microRNAscope assay. Again, the same three applications where ASOs have been developed for inhibiting expression of certain cancer-promoting genes. For neuroscience, they have been in development for treating neuromuscular and neurodegenerative disorders. And for immunology, again, for treating autoimmune disorders. So an example again here from one of our customers, they designed an antisense oligo therapy and specifically treated muscle tissues. We have here a vehicle-treated panel and the actual treated tissue for mouse cardiac muscle. And using our ASO-specific probe, they were able to show that they were successfully able to treat this particular tissue using their ASO. Similarly, in the mouse quad tissue, they were again able to show that the treated tissue showed very high expression of their ASO probe. So this particular assay has been very successfully used even in its early days to assess the efficacy and delivery of ASO and siRNAs in target tissue. So overall, the RNASCOPE technology, which includes all these three assays, have been applied quite widely in the recent years in preclinical models, but with very strong clinical applications to understand the efficacy, biodistribution, and the safety of these therapies, which is extremely important going forward before translating into actual clinical treatment. So over the last few years, our technology has been widely applied. We have seen a lot of growth in the number of publications. We have over 2,700 publications as of a few months back. Furthermore, the incorporation of the RNASCOPE technology into single-cell RNA sequencing workflows has demonstrated the rapid growth of our assay in recent years. And as I mentioned, we have been applied in different research areas like cancer, neuroscience, infectious disease research being our top areas of publication. So in summary, our ACE technology is an ideal solution to detect therapeutic or target gene expression and vector sequences in individual cells within the tissues with morphological context, and to address critical questions on efficacy and safety behind nucleic acid and cell-based therapies. So it can be used to visualize in vivo delivery of therapeutic targets, just viral and non-viral vector expression, and localization of the transgene expression in a specific cell type. So overall, all of our three assays that come under the broad umbrella of RNASCOPE technology have been used for cell and gene therapy applications. With that, we will take any questions at the end of our presentations, but now I'll hand the platform back to Rhonda. Thank you so much.
Thank you so much, Anushka. That was a fantastic presentation. And yes, please, if you have any questions, please pop them in the chat. I put a little reminder in there. We will have Anushka and Bree at the end of the presentation today getting ready. So with that, I'm handing it over to Bree. Gabriella Perell, she's our product manager for cell and gene therapy for the BIO-TECHNE group. And if you're ready, you may go ahead and start.
Oh.
We can hear you now, yes.
All right. Well, thank you. I appreciate the offer to come and speak today. So I'm going to talk a little bit about some tools and reagents that we've used to kind of enhance the workflow for cell and gene therapy processes. So about 2 years ago, we started our cell therapy initiative at BIO-TECHNE. And if you're not really aware of BIO-TECHNE, this is sort of an all-encompassing company that has several different sub-brands that includes R&D Systems and ACD and Protein Simple. And all of these sub-brands have really strong areas that were identified to really benefit cell and gene therapy tools and reagents. And so this initiative was started to really accelerate the research within this space. Cell and gene therapy has really expanded in the last several years. This is a table sort of highlighting not only the variety of different cell types that clinical trials are going on, but also the breadth of different disease states. You can see quite an array of different areas that cell and gene therapy are targeting. With oncology, at a significantly higher amount. But the breadth of this space really shows the need for robust and strong cell and gene therapy processes, and with that, the manufacturing processes and all of the different reagents and tools that go along with that. And there have been some hurdles that this space is really identified and is addressing. And some of these go along with supply chain and consistency. So having reproducible processes in place that really minimize the amount of time that it takes to get to the patient. And along with that, having high quality reagents. So having GMP grade materials that you understand the raw material sources and that they're complying and being able to be used in your final product. So we really took these hurdles to heart and really are working to address these difficulties within this space. So at BIO-TECHNE, we have the largest portfolio of GMP proteins and small molecules. So we have that historical quality that we can refer to and really understand and identify and use to have consistency along the way in terms of our proteins and small molecules. We have GMP grade manufacturing and expertise within this space. We are actually opening up a brand new GMP facility that will help with supply chain demands as well. And we're also continually optimizing our reagents and protocols so that these processes can not only be used at the research and development scale, but can be scaled up readily and easily into preclinical and clinical stage. We also wanted to have platforms that are flexible, that can be used within any process. And so it's easy to adapt and implement. And that goes along with our ability to customize and make these fit a specific process. And we also have reagents that can translate from development into preclinical with both RUO and GMP reagents. So this is our immune cell therapy workflow that we look at. So this is starting from the beginning with the leukapheresis packs and how this process really plays and how we impact each of these processes. This then gets into a cell selection operation, and then in gene editing, where we have B-Magen tools to help with the editing. We have cell expansion and activation, which I'll mainly talk about today, and then formulating that material and getting that back into the patient. So we really are working to drive specific tools for these specific areas. But today I'll focus primarily on cell expansion. And some of these tools that we use are shown here. So that includes our GMP proteins that we have created specific ProDots that I'll talk more in depth for closed processing systems. An innovative hydrogel that's part of the Clouds brand that can expand and activate cells. That work with our optimized accelerate medias. And we also have gene editing tools as well that today I won't speak about. So part of addressing some of the hurdles within the space within cell activation was going to look at the methods that are used currently. So right now we use typically, the majority are using magnetic-based protocols. While these work to expand cells efficiently, there's some difficulties in removing the cells, or removing the beads, making sure that all of the beads are taken out. And so we really tried to develop a novel tool that would minimize the cell processing and the cell washing to optimize this process. And that's where our Clouds microspheres come into play. So these are hydrogels that are functionalized with antibodies of interest that will activate the cells. So we have certain Clouds reagents for T cell activation, and that uses CD3 and CD28 antibodies. We have NK cell activators that uses CD2 and NKp46. And what's really unique about these hydrogels is that they readily dissolve with a specific release buffer in less than 5 seconds. So you can see here, these are the Clouds particles. And I'll play a short video. You can see that as the release buffer is added, those reagents completely dissolve away, and you can readily wash them away from your cells of interest. Right now, we have a variety of different kits available, both in GMP and RUO. I want to talk a little bit about the process that we have used for NK cell work and how we are optimizing this protocol with our Accelerate NK media. So shown at the top is the protocol for a 14-day cell culture activation. And this uses our R&D systems, cytokines, IL-2, 12, 18, and 21. And you can see the full protocol. I won't go into all the details, but this is optimized with our Accelerate media that is a serum-free media that is works just as well as serum-containing suppliers. This is a xeno-free version as well. So a little bit of data to highlight. After the 14-day activation, we see an expansion of those NK cells to about 170-fold. This is somewhat dependent on the donor. And then we also see a viability of those cells around 85%. I want to kind of highlight the PRODOTs as well, so kind of changing gears here. The proteins that we have historically made, we have modified these to what we call PRODOTs, which are these spherical lyophilized balls of cytokine that are within enclosed bags shown here. Right now we have IL-2, 7, and 15 for immune cell culture, and we're working to expand this portfolio into other cell culture-focused areas. So there are some of the challenges that have been identified within this space in that cytokine addition into your media is not always a closed process, and having a scalable and flexible system had been identified as an issue that we took to heart and focused on. And these PRODOTs are automatively compatible. And we've also identified lot-to-lot consistency that is very high. These are animal-free under GMP-controlled conditions. And you can see here the bag on the right can contain up to 25 mils of media to reconstitute the cytokines. Each sphere or protein ball is 5 micrograms of protein, and dependent on which cytokine you have, we have different number of proteins within each bag. These are sterile and QC tested up to USP 71 standard as well. So I just want to show a short video on how the proteins here you can see dissolve rapidly. So we're adding in a media. I'll just let you watch for a minute. So you can see that the media dissolves the protein within a minute. We see complete reconstitution. And then we also have comparability data to our standard proteins as well. So we have tested this with our T-cell clouds activator with T-cells and our Accelerate media, with our IL-2 standard protein and then our IL-2 PRODOT. And we see expansion basically identical, as well as in terms of phenotype, both CD4 and CD8 similar as well, indicating that these are equivalent to our regular GMP proteins. So that's all I have today, just a kind of a brief overview of the different tools and reagents that we are focused on bringing to market to really drive and help this space. So appreciate the time and love to hear any questions you have.
Thank you, Bree. That was a fantastic presentation.
Hi, Rhonda. Hi, Anushka, and hi, Bree. Thank you so much for the presentation today. It was fantastic. And everybody else, thank you so much for joining us for the presentation. And we truly hope you learned something new from our discussion. And if you're interested in learning more, please do reach out to us. We'd be happy to continue the conversation. And once again, thank you so much for your time today.